Hi class, and welcome to the second screencast in our unit on molecular genetics. So we've already learned all about the structure and function of DNA and how DNA gets replicated in the nucleus so that a new cell can get a whole copy of DNA. But now we really want to come to the crux of the situation here. How does that DNA that we learned about make us into unique individuals? How do we go from DNA to all of the various proteins that make us who we really are and allow us to function? Well, this brings us to the central dogma of biology. It's called the central dogma because everything in life is really centered around the idea that we start with DNA, and DNA makes RNA. This is sort of like an intermediate step, and then RNA is turned into protein. So all those proteins, again, make us who we are. So we're going to learn about two very important steps. The first step is what makes DNA into RNA, and this is called transcription. And this happens in the nucleus. And so we'll take a closer look at this process later in the video. And the second process is called translation, and that takes RNA and turns it into the final protein product. And this occurs in the cytoplasm on the organelle called a ribosome. Now, another really good thing about this video here is we're bringing a lot of information that we've learned all throughout the year together in sort of a culmination here, learning about how proteins get made. So we know what a protein is, we know what DNA and RNA is, we know what a ribosome is, and so now we're going to put it all together. So remember, we're making a protein, and proteins determine our phenotype. Remember, phenotype means what we look like, how our bodies function, etc. And we learned all about proteins early in the year. They have so many different functions. They act in our immune system as antibodies. They act as enzymes. They have lots of different functions for every form of life. So here's just a very good overview diagram that I need you to draw in your notes. I need you to draw this for me. So remember, we already learned about this, right? So I'm just going to sort of draw a circle that demonstrates where the nucleus is. Okay, so this blue circle, this is your nucleus. We learned last week about how DNA is replicated with DNA polymerase into another DNA molecule. And we're going to learn in this video how we take DNA and how we turn it into RNA. Remember, that's called transcription. And the enzyme that does that is called RNA polymerase. So not too hard to remember that enzyme. And then RNA is going to travel to the cytoplasm. So out here is the cytoplasm. And translation is going to turn that RNA into a protein. Now, can you remember what you think these little subunits are? What are the monomers of proteins? See if you can remember that. We'll come back to that. So draw this in your notes. Okay. So I've told you all of this, but how do our cells know how to read the DNA? How do we start with that DNA? And the answer is the genetic code. So I'm going to spend a few minutes here talking about the genetic code. This is so important to understand. So the DNA language, and we already know this, is written in something called nucleotides. Nucleotides are the monomer of DNA. And it gets transcribed into the same language of nucleotides, but it gets transcribed into RNA. So something that gets transcribed, you're just taking, a language to a similar language. So for example, we're taking the DNA nucleotides of TAC, etc., and we're transcribing them into the RNA nucleotides of AUG, GGC, UUA. Now you might be wondering, how did I know that that was the RNA that I was supposed to make? Complementary base pairing. So remember, T always pairs with A, and A always pairs with T. But remember, earlier in the year we learned that RNA does not have T, RNA has U. So A is going to pair with U, C is going to pair with G, and so on. It's the same concept as DNA replication. It's complementary base pairing. So now we're done with transcription. We have our RNA. Now we have to get to the protein. So this is a little bit more complicated. The RNA nucleotides are going to be translated. Now, when you hear the word translate, you should be thinking about a new language. You translate from Spanish to French. It's a new language. The new language we're talking about is the language of amino acids. Remember, amino acids are the monomers that make proteins. So that is translation, and the amino acids that would come from this are methionine, glycine, and leucine. Now, how did we know that those were the amino acids that were supposed to come from that RNA? Well, we know that because of something called a codon. 3 RNA nucleotides make up one codon, and one codon specifies one amino acid. So let's go back over here, and I'm going to underline each codon. So AUG is a codon. GGC is a codon. and UUA is a codon. So we're going to see that AUG is going to tell the cell to bring methionine, GGC tells the cell to bring glycine, and UUA tells the cell to bring leucine. And we're going to do lots of practice with this genetic code in class. So some more about the genetic code. It's also called the triplet code because 3 nucleotides make up 1 codon. There are 64 total RNA codons, and I do need you to know some of this information. So remember, codons are actually RNA, not DNA, but RNA. 61 of the codons correspond to distinct amino acids. Now remember, there are only 20 amino acids, which you should be thinking, okay, so then more than 1 codon can code for 1 amino acid, and you're exactly right. AUG is an important codon you have to know because this is always the start codon. Every single protein starts with AUG, and the enzyme, or the protein, sorry, the amino acid that it brings is called methionine. That starts it all. And then the other 3 codons are called stop codons, and this signals the end of making that protein. So here is the genetic code in a chart form, and we're going to learn how to read this in class and use it in class. But basically what you see along the edges here, this is the first base of the codon, this is the second base of the codon, and this is the third base of the codon. So let's start with that AUG. Remember I said that was a really important codon to know. So if we start over here with the first base, we find our A, and then we go over to our second base and we find our U. So here's our A, and here's our U. So right now we're in this box right here, but now we need to go over to our third base all the way over here and find our G, which is right here. So we bring that all the way over and look at what we come to. We come to methionine. So what I want you to do right now really quickly is I want you to see if you can figure out what amino acid CCU would code for, and write that in your notes and practice using that genetic code. Now the really cool thing about this genetic code is that it is universal across all life forms. So little tiny bacteria to humans to plants, all life forms use this same genetic code. AUG always codes for methionine. GCA always codes for alanine. The same codons and the same amino acids. I just think that is incredible. Okay, so the best way to get into the groove of the genetic code is to practice. So I want you to transcribe and translate this DNA sequence into RNA, and then I want you to transcribe those codons into the amino acid sequence using the genetic code that I showed you on that last screen. So you're going to have to pause your video on that screen to do that practice. Okay, so now let's take a closer look at that first process of transcription. Remember, this is happening where? In the nucleus. Very good. So this is happening in the nucleus. So what has to happen first is the 2 DNA strands have to separate from each other because transcription only uses 1 strand of DNA, not both. And that 1 strand that is used is called the template strand. And so this is going to be used as the template or the pattern to make that RNA chain, which uses that base pairing. So A always pairs with U and G always pairs with C. And the enzyme you have to know that makes that reaction happen is called RNA polymerase. So here's a very quick look at transcription. What we're looking at here, this is the gene. So this is the DNA. And this RNAP, this is RNA polymerase. So RNA polymerase is going to just sort of open up the DNA and it's going to read it and make an RNA molecule out of it. So here's how that happens. RNA polymerase unwinds the DNA and it's making this new RNA molecule from that template. So now we have this complete RNA molecule in blue, and now that's going to go to the cytoplasm for the second process, which remember is called translation. Okay, so let's go back to this. So we've got a whole bunch of DNA, right? We've got a whole bunch of genes. The question you might be thinking is, do all of our cells transcribe every single gene that we have all the time? So does every gene get made into RNA all the time? And the answer is no, and this is another really cool part about life. Gene expression is highly regulated so that only certain specific proteins are made and only when they're needed by that cell. So what this allows for then is cell specialization. So this makes, say, a skin cell different than a nerve cell, even though the nerve cell and the skin cell actually have the same DNA. They have the same genome, all of your cells have the same genome, but only certain genes are transcribed and translated because that cell only needs certain proteins. So that's a really fascinating thing about life. Okay, let's talk about RNA for a second. There are 3 types of RNA. Messenger RNA is the type that we've already seen. That's what takes the message from being transcribed in the nucleus down to the cytoplasm and to a ribosome. So that's what we just saw being made, messenger RNA. Now, ribosomal RNA is a special type of RNA. Will associate with proteins and it makes ribosomes. So this here is an example of a ribosome. So yes, ribosomes are actually made out of nucleic acid. And the last type of RNA is called tRNA, transfer RNA. This is going to be very important in the process of translation. It brings the correct amino acid to the ribosome during translation to match with its corresponding codon on the mRNA. So keep in mind all 3 of these types of RNA. So here's a very good overview of all of these processes I'm talking about. So we start with DNA, we go to RNA through transcription, and remember I just told you it's mRNA that's being made. That's going to take the message. It's a messenger. It transports it to the cytoplasm to make polypeptides, to make proteins. So here's your RNA molecule in blue. This little shaded guy back here, that's your ribosome, which remember is made up of ribosomal RNA. And then these funny looking guys that kind of look like clovers, these are your tRNAs. Remember, tRNAs bring the amino acid. So this little ball here, that's your amino acid, and it brings the amino acid to the correct codon. So the codons are these little 3 nucleotide base parts here. And then the amino acids are going to bond together and eventually make a final protein product. So that's a good overview. I highly, highly recommend drawing that in your notes and labeling it as much as you can. Okay, so let's take a closer look now at translation. As we just saw, ribosomes are really what is going to build the polypeptide. So translation occurs on the surface of the ribosome. Ribosomes have 2 subunits, a small and a large subunit. Each subunit, like I said earlier, is composed of both ribosomal RNA and protein. And finally, ribosomal subunits only come together during translation. If a protein does not need to be made, ribosomal subunits are just sort of apart from each other. But then once that messenger mRNA comes in, the 2 subunits grab onto the mRNA and they start reading the mRNA. So they've got binding sites both for the mRNA and for the tRNA. Okay, so this is one of the last slides here, but I'm going to spend a lot of time on it. You also need to draw every single thing of this in your notes and label as much as you can. So again, starting from the beginning, here is our mRNA molecule in this light green color. Remember, this has come from the nucleus and now it's in the cytoplasm and it sits in between the 2 ribosomal subunits. So here's the large subunit and here's the small subunit. So now the really important part here is the genetic code, right? We have these codons on the mRNA. So for example, this codon is GAU. Well, that tRNA that's going to match up with it is called CUA. Now, there's another really important part of this tRNA, and that's called the anticodon. So that's what pairs with the codon. So that's an anticodon, and that's an anticodon. Notice again, it's just base pairing. So A is going to pair with U, C is going to pair with G. So that tRNA has an anticodon on one end, and then on the other end, it has that amino acid. So this one is carrying phenylalanine, for example. This one is carrying ASP. This one's carrying lysine. Each tRNA is going to carry a different amino acid, and therefore it's going to have a slightly different anticodon. Okay, so we have the tRNA come in, it binds to the codon, and it's carrying its amino acid. It's going to sort of drop off that amino acid onto the growing peptide chain, and then once that tRNA is empty, it's going to go back and it's going to repeat the cycle. It's going to pick up another amino acid, and it's going to come back when it's needed next, whatever the mRNA says. And this is how our protein is actually made. It's just amino acids being put onto the end of the chain. And so, at the end here, we need to start to build our protein, because remember we learned about earlier in the year, the primary structure is simply just this sequence of amino acids. But then we get into secondary structure, so those amino acids start to build on each other, and finally we get the quaternary structure. So that is the final protein, and that's what makes us who we are, that final quaternary structure. Now I have 2 questions to leave you with, and I will check these in class.